Hello, grade 9. Today our lesson is about enzymes. The key concept is enzymes are catalysts for chemical reactions in living things. As you see here in the figure, it's a plant called Venus flytrap. It can digest insects and animals like frogs. How this happens? Just how can a Venus flytrap digest a frog? It happens through the action of proteins called enzymes. So enzymes help to start and run chemical reactions in living things. These enzymes are catalysts, and although catalysts take part in a chemical reactions, catalysts are not considered to be either reactants or products. They just run or speed the reaction. Okay, because the catalysts are not changed or used up during a reaction. Now, there are some functions for the catalysts. Catalysts are substances that speed up chemical reactions. They decrease activation energy and increase reaction rate. If you look at the figure below, we can see here at normal reaction, and this line, you can see we need more energy for activation of the reaction. While in the presence of catalyst or enzyme, the catalyst will increase the rate of the reaction and we need less energy. So the activation energy here is lower with the presence of the catalysts. So, if you ask this question again, describe two functions of catalysts in chemical reactions, the answer should be catalysts decrease activation energy and increase reaction rate. Enzymes are types of the catalysts. Enzymes allow chemical reactions to occur under tightly controlled conditions. So enzymes are catalysts in living things, and they made of protein. Most of enzymes made of proteins. Now, they are involved in almost every process in organisms. Like what? Like breaking down food, building some molecules like proteins. We need enzymes. So in most of the processes that happen in our bodies, enzymes are needed. They are involved in all the reactions. For example, the amylase enzyme, which exists in the saliva, the liquid that lubricates the food in our mouth. This amylase enzyme can break the starch into simpler sugar, as if you eat a piece of bread is a starch. Your body should break it down to smaller, simpler sugar by the amylase, so it can be absorbed by your body. This reaction occurs up to 1,000,000 times faster with amylase than without it. Okay, so we saw here that the reactions are speeded up by the action of the catalysts, which are the enzymes. Now, enzymes function must have certain conditions to function properly. Any disruptions in the homeostasis can prevent the enzymes from functioning. A reminder for homeostasis. Homeostasis is the internal condition of the body. Each body should keep its internal condition normal. If there is any disruption like increase in the temperature, like you have high fever, this will make disruption in the homeostasis and this will affect the enzyme functioning. So, enzymes function best in a small range of conditions. Okay, changes in temperature and pH can break hydrogen bonds. As we learned before, enzymes are proteins. Well, proteins have folded structure. If there is any change in temperature or pH happen, the folded structure will be changed because the hydrogen bonds will break down. Okay? So this is one reason why a very high fever is so dangerous to a person. Also, in addition, a change in pH can affect the hydrogen bonds in enzymes. Many enzymes in humans work best at the nearly neutral pH that is maintained within cells of the human body. So, as a result, enzymes function best at certain conditions, small range of conditions. So, the enzyme's function depends on its structure. If any change happen in the structure of the enzyme, then the shape of the enzyme, this will affect the function. Now, this question, how does the structure of an enzyme affect its function? And the answer is, if the structure of an enzyme changes, its function will change. And what will cause the enzyme to change its structure? Any changing in the conditions, like changing in the temperature, changing in the pH. So an enzyme's structure is very important because it allows only certain reactants to bind to the enzyme. The structure is very important. If you look here, the enzyme, which is shown here, this one is the enzyme, have a structure, okay? It have active site on it, all right? This enzyme can bind to certain reactants, which we call them substrates, okay? They bind to the enzyme at certain place called the active site. For example, the amylase only breaks down starch. Therefore, starch is the substrate for amylase. Substrates temporarily bind to enzymes at specific places called active sites. In the same way that a key fits into a lock. It's like a key that fits into a lock. So substrates exactly fit the active site of enzymes. This is why an enzyme structure changes. It may, if it changes, it may not work. It's exactly like the key and its lock. So this idea of enzyme function, which is called the lock and key model, is shown below. Look how does it work. The lock and key model. This explains how the enzymes function. Now, as we said, the enzyme here, this is the enzyme, have active sites, okay? And this, here are the substrates, the reactants. They will bind to an enzyme at certain places. Both of them will bind here. Then the enzyme brings the substrate together, as you see here, like the key and its lock, exactly. Okay, and weakens their bonds. The bonds between the reactants will be weaker. After that, the reaction will happen, the catalyzed reaction forms, and the product is released, new substance, from the enzyme. Did the enzyme change? No, the enzyme stay the same. It only take a part in the reaction, but it doesn't change. Let's see these questions. How does a catalyst affect the activation energy of chemical reaction? As we said, catalyst reduces the activation energy or decreases the activation energy required to start the reaction. Describe how the interaction between an enzyme and its substrate changes a chemical reaction. An enzyme brings substrate closer together so that they can react. Okay, and it slightly weakens the bonds within the substrates by changing the shape of the molecules. Thank you for listening and see you tomorrow.